I'd like to start off by acknowledging the land that I am sitting on and the land that my museum is on. The Burke Museum and myself stand on the lands of the Coast Salish people whose ancestors have resided here since time immemorial. Many indigenous peoples thrive in this area, alive and strong. So, like Jade said, I'd encourage you to find out what indigenous lands you are occupying right now. So, I'm going to talk a little bit about my job and how it impacts the greater view of paleontology and the bigger field of paleontology, because science is all connected and it goes back and forth between the different teams and the collaborators. And the way we collect data is very complicated, and managing data is very important if you're doing scientific experimentation. There's a lot of lack of documentation in science as well, which can be a really big problem. So I'm going to talk about some of the dangers of how we get fossils and how my job can help or harm science and what we are doing with fossils for an eternity. If I can figure out how to move my slides, there we go. So I'm going to start by pretending that we're tourists and we're walking into the Burke Museum. We're going to have our normal experience that most of us have had when we go into a museum. So at the Burke Museum, we go up a giant elevator. It's the size of giant dinosaurs because we have to bring our dinosaurs into our museum. And the doors open and we look out into our fossil gallery. And right off the bat, we see this big old stegosaurus. And got to run over to the stegosaurus. It's a charismatic giant dinosaur from the Jurassic Formation. Many people see those big old plates on the back and it's very recognizable. It's so cool. And it's being chased by a theropod. It's got an Allosaurus coming after it. And right behind it is a T-Rex skull. So you immediately go past those Jurassic dinosaurs to go look at that T-Rex. The picture right there is the Tufts Love Tyrannosaurus Rex. That is the T-Rex skull that is on display at our museum. It was prepped in the museum, it was collected for the museum, and it's the best T-Rex skull in the world. That's no little flex. We have every single bone of the skull, including the ear bones. And that was beautifully prepared by both volunteers and staff at the Burke Museum. Gorgeous, gorgeous fossil. But back behind it, I see some other big old skeletons. Oh my gosh, what's that over there? There's a whole Ice Age display with huge skeletons. And I can stand underneath this mammoth and look up at this huge mammoth. And it's so cool. Wow, this is so cool. But there's archaeology on the other side of the building. Let's go and see archaeology. And that is our experience of a lot of museums. We run in, we see the big cool things. We go, wow. We take a selfie and then we go see the next cool thing. How long did I just spend looking at these giant skeletons? A couple minutes? A couple seconds? Think about how long you look at skeletons when you're at the museum. A minute or two? That skeleton that you're looking at and you're enjoying for 2 minutes took hundreds, if not thousands of hours to be presented to you in that form. A lot of things happened to that fossil just to give you that little blip of joy. What was the point of that? Why did we spend all that money and time to give you a skeleton that you're not going to enjoy for a whole lot of time, that you have no idea what it took to give you that skeleton? Well, there's a couple reasons. One, we do want to give you that little blip of joy. We want you to go, wow, I'm under a mammoth. That's so exciting. That's so cool. We want to be able to share fossils with the public. We want to know our past and understand how we got here and the evolution of life and the different environments that the world has gone through. But now that skeleton is out of the rock and we can study it for science. So we've got two goals and two reasons we're making these giant skeletons and spending hundreds and thousands of hours on these skeletons. We want to share them and we want to study them. So let's jump into the study. We have our mammoth skeleton out on display. Let's go measure. Let's go take samples. Let's go learn about how it lived and died and what it ate and what it lived with. What environment was it in? How did it see? How did it mate? Did it migrate? Let's go find out all these things about the animal now that we have this skeleton out. And that's how I wanted to do paleontology. That's how a lot of people want to do paleontology. You want to be a researcher. You want to be a doctor at a college and you want to do research and you want to become famous and be on TV and Nova specials and PBS and you want to talk about all this cool science. I wanted to do that. So about a decade ago, I went into grad school to get my master's degree in geoscience. And when you go in your master's program, you get to pick your first big research project. And I was so excited. And when I get to my college in Fort Hays State in Kansas, they had a fairly good size of Miocene rhinos. So 15 million-year-old-ish rhinos from Kansas. And I have a photo of the rhino up on the screen right now. That is Teleoceras, um, and that is a rhino that lived in North America. There were several species, and they looked kind of like the dachshund of rhinos. They had short, stumpy legs, long bodies, and they also lived at the same time with multiple other species and kinds of rhinos. So I'm looking at all these really cool rhino fossils, and I'm sitting here thinking, what did they eat? Because I like food, and there's a whole bunch of rhinos here. What kind of food did they like? How did they all survive in the same environment together? So I had to pick my project. I picked my animal. I liked these rhinos, and then I wanted to study what did these rhinos eat. And so I'd come up with my plan and my research and my goal, and I needed to pick a technique. How do I find out what animals eat? So I did some research. I read some papers. I Googled and went down some Wikipedia rabbit holes. How do we find out what animals ate? And I came across a technique called microwear. It's like, great, got my technique. I'm going to find out what these rhinos eat. I'm going to publish it. We're going to figure out so many mysteries about these little dachshund rhinos from Kansas 15 million years ago. I skipped a slide, sorry. So, microwear. How am I figuring out what these animals ate? Well, the theory behind this is we are looking at the surfaces of teeth under microscopes at high magnification. And when we blow up the surface of that tooth and we look very closely, we can see lots of scratches, those long lines, those depressions in the fossil, and we can also see little holes or pits. What's causing those scratches in those holes and pits? Well, the thought is it's the food interacting with the tooth. So as this rhino is eating grasses and it's chewing those grasses, there are little crystal parts of the plant that are grinding up with the tooth. Most plants have tiny silica crystals that make up the structure of the plant. Silica is glass. These little silica structures are called phytoliths. You can identify groups of plants based on the shape of the phytoliths, and they make the structure of the plant. And so it's thought that those little glass particles are interacting with the tooth. And you can look at how many scratches there are versus pits and the shapes and the sizes and the distribution of them. And you can compare that to modern animals that you know the diet of. So we can go look at modern rhinos, black and white rhinos from Africa who have different diets. One is a browser and likes the low shrubby bushes, and one's a grazer that likes grasses. So we can go look at their teeth and say, yep, that one ate grass. This is what the scratches and pits look like. And we can look at one that ate bushes and say, these are what the scratches and pits look like. Great. We're going to find out what these ancient rhinos ate by comparing them to modern rhinos. This sounds flawless. This sounds great. I'm going to learn so much about what these little chubby short rhinos were eating. Whenever you think something's going well and it's going easy, that should be a warning in the back of your head, because I was soon thrown a big curveball. I picked my technique. Well, when you're doing science, science has to be repeated. It has to be replicable. I have to be able to say, I did these steps, and other people are supposed to follow those steps if they want to do what I was doing. If they want to compare results, you have to do the same thing as someone else. If you change any of the steps of the process, you're not doing the same thing. It's not really comparable to the previous data that you've been collecting. So science has to be repeatable, and you have to do the same thing as everyone else in order to have that same kind of set of data to compare. So when I pick microwear, I have to do what the methods paper says. I have to follow every step. I have to do what they say, the way they say, or else whatever results I have aren't really proper to compare to what they did, because I did different things if I pick a different material. This isn't always great. There are a lot of chances, or there are a lot of places in life where you're told to do something, you're handed something, and you're said, use this. And we don't always ask, why am I using this? And can I use something better? And what else is out there? We don't always get curious about the things that we're handed. We just say, yep, okay, I will use this. And up until the point of my master's degree, I had been doing that exact thing. I would go to museums, I'd be an intern, I'd be a field worker, and I'd be out in the Badlands of Wyoming digging up Jurassic dinosaurs like that Stegosaurus, and I would be handed a bottle of glue. And now this is a side tangent. Glue used to refer to animal products, and someone older than me might yell at you for using the word glue. For today's discussion, glue is the sticky things we put things together with. A lot of glues don't have animal products anymore. I won't yell at you if you say the word glue. So we're going to use the word glue. That's sticky stuff. We're not going to worry about what it was made with right now. But I'm handed different types of glue at every museum. At one museum, we're digging up Stegosaurus, and I'm handed a bottle of super glue, and I'm told to dump super glue on the fossil. And don't worry, we're going to use super glue when we go to the lab to get the fossil out of the rock. Okay, cool. That's what you use. I go to another museum to work, and we're digging up Diplodocus, another Jurassic dinosaur. And I'm handed this bottle of clear liquid called Boutvar B76 or something. It has a number, it's got a whatever weird name. I don't remember what the name is. I dump it on the fossils because that's what I'm told to do in the field. I'm just a little field assistant. I like digging dinosaurs. I don't know what I'm doing. I'm not questioning. I'm just doing as I told. I'm not curious. I don't care about the glue. I want to dig up the dinosaur. That's the cool thing. The glue is just holding it together so that I can play with this fossil. I go to another museum. They hand me another glue in the field. It's called Vinax something. I don't know what any of this is. I just want to play with the dinosaur. I go into their lab to get my cool Diplodocus fossil out of the rock, and they give me Elmer's glue. White Elmer's glue, the same craft glue you use in middle school, and you put on your hand and you peel off. And I'm told to glue an Allosaurus arm bone together with Elmer's glue. Oh, that sounds a little weird to me, but sure, okay. It's whatever you tell me to do to your fossil, I will do to your fossil. So up until my master's degree, I'd never questioned the materials I was given. I just did as I was told. But things were changing now. This was my research. This was the paper I had to write. This was how I got my degree in the Masters of Geoscience. This is how I got into my career, my dream job. This is how I became a paleontologist. I want my research to be good. I don't want to do something bad. I don't want to be wrong. I don't want to publish a paper and then have a bunch of people say, you did that wrong. I want to make sure I did this right. I want to dot my I's and cross my T's. I want to know everything about what I'm doing so I know that no one can say you did that wrong. I want to say, this is why I did it, and this is why I didn't do another way. So I'm starting to question what I'm doing. I'm getting anxious that I'm going to screw up. I'm thinking about everything that I'm doing. I'm overthinking what I'm doing. I'm going down Google rabbit holes. And I found out some really bad things. So on the left is a jaw of a Teleopterus. That's a rhino jaw. And you can see it looks kind of like it's got plastic and crystal growth. There's some shiny bits. It's yellow. It's kind of caked and gunky. I can't really see the surface of the tooth. And I'm supposed to be looking at those microscopic bits on the tooth. Well, if the fossil has glue on it, like this fossil on the left, and I am using glue correctly, that is an animal-based product on the left, I gotta get the glue off. I gotta get to the surface of the tooth or I can't do my research. So this method has a way of removing the glue from the tooth so you can get to the tooth surface. Great. I'm gonna do what they said, the way they told me to do it, so that I can replicate their study. So this glue on the left is what you call shellac. 100 years ago, when we started preparing fossils and taking them out of the rock, shellac was a very common product. You poured it on the fossil, it sunk into the fossil, it got hard, made your fossil stick together, looked great. Well, over time, shellac turns yellow, it expands, it contracts with heat and humidity. There's dust on that fossil and it's expanding, it's incorporating that dust. It's something that we call a non-archival product, which means it goes bad, it turns yellow, it off-gases chemicals, it reacts with the environment, it degrades over time, it starts to fall apart. 100 years ago, they didn't know that. They weren't thinking ahead. They weren't thinking about this goop that's now on the surface of this tooth. 100 years ago, they didn't know what microwear was. And today, even now, when I'm applying materials in glue to fossils, I don't know the science that they're going to come up with in 100 years. 100 years ago, they didn't know we were going to come up with CT scanning. So they were dumping all sorts of things inside of bones because they didn't know we were going to go be cutting up those bones. So 100 years ago, they weren't anticipating the change of science and the development of new methods. They were using what they had. They might have not been thinking about it that hard, and they were keeping their fossils together. So I was given a shellacked jaw. I was lucky enough to know that there was shellac that was on it, but other than that, I had no idea how that fossil had come to me. I don't know how they got the rock off. I don't know if they scrubbed it with a toothbrush. All I know about this fossil is it has shellac on it. I need to get the shellac off, and I need to look at those microscopic parts of the tooth so I can figure out what this rhino ate. So I follow directions on how to remove shellac from this fossil. Huh. How do we remove it? We dump a bunch of shellac remover, which makes sense in your head logically. Okay, shellac remover, remove shellac. We dump that on the tooth. We let it sit for a while, and then we scrub it off with a cotton Q-tip. Our tooth is clean, it's beautiful. We can go do our science. Hold on. I'm getting some uncomfortable vibes right now. I'm worried. Am I doing something wrong? Is someone going to yell at me? I am scared about my thesis paper. So I Google, what is cotton? I don't know what cotton is. Cotton's a plant, right? Well, hold on. These teeth are supposed to be recording plant material, right? Is this cotton ruining my tooth? I don't know. I have to go find out. So I'm Googling furiously about cotton. Well, good news. First thing I find out, I'm so relieved. Cotton doesn't have crystal structures. It doesn't have phytoliths. It's the fluff around a seed. Good. I don't have to worry that I'm scrubbing my tooth with those little silica particles and making new ones or changing the old ones. We're all great. But hold on. That fluff is attached to a plant that has phytoliths. It has those silica crystals in that plant. And while that cotton is growing in that field, those plants are decaying. Those glass crystals don't disappear. They're incorporated in the soil. They're blown around on the plants. And then all those plants with that cotton fluff are harvested. They're dry. Silica particles are going everywhere. Leaves are going everywhere. Sand from the soil is going everywhere. That cotton is getting contaminated with all sorts of things from the environment that could affect what my tooth looks like. Oh no. Have I been scrubbing my teeth with silica? I have no clue. How does this process? I have to go find out now. I'm going deeper into this rabbit hole, and I'm going farther away from figuring out what my rhinos ate, because now I'm trying to figure out how am I screwing up my rhino and my thesis and my ability to get a job and my degree, and it's a panic spiral. And it's my worst fears ever. On the left is a really close-up microscopic view of a cotton Q-tip that you'd go get at Walgreens to clean out your ears and clean off your makeup brushes. You see the fibers of cotton. Cotton is made out of cellulose. It's a plant fiber. It's really soft. It's fine for fossils, as far as we know. There's no sand grains. That looks great. Cool. My cotton Q-tip from Walgreens is great to scrub my teeth with. Until I look at the next Q-tip that I pull out of the box. These aren't my photos. This is hypothetical. I pick out my next Q-tip from the box. And on the right, that's a sand grain. That's a big chunk of glass that's in that Q-tip. And that's because not all cotton fibers are the same. Some of them are contaminated with the particulate that was in that harvest. You're playing Russian roulette when you're using that Walgreen Q-tip. I don't know if that Q-tip had silica in it when I went to go scrub that tooth. Maybe it did, maybe it didn't. I have no idea. Maybe I took that clean cotton Q-tip that didn't have any silica and I set it on the table in a museum collection full of dust and dirt. And then I picked it up again and it had that dust and dirt on the table that I couldn't see. And then I scrubbed my tooth with that dust and the dirt that's probably gouging my tooth in microscopic ways that I don't even know. I'm starting to really panic spiral. And then there's a light, there's a hope again. I'm learning about this cotton and oh my gosh, okay, it's the United States, so we have all sorts of regulations about what we're putting in the cotton and how we're communicating products to customers. There's a whole system for cleaning and grading cotton. Yes, okay, we're getting somewhere, okay? So I know on the left here, there's leaf grade, trash, extraneous matter. Companies know there's goo and there's gunk and there's trash, and they call it pepper trash, silica, rock in their cotton. Cool. They know how to clean it out. There's very standard protocols for purifying cotton, and there's even names to indicate that you have purified cotton that has no silica or contaminants in the environment. And the way that we do that is we have the US Pharmacopeia, I can't say that word, convention, or USP, that has a set of regulations to purify and make sure that that cotton is clean. And there's a lot of uses for clean cotton, right? You need sterile cotton for medical practices. You can't use your Walgreens Q-tip to clean a microscope because that chance of contamination could scratch your microscope. So these USP cottons have nothing in them. They're clean, they're pure. You can clean your microscope. You can clean delicate instruments. You can clean fossil teeth with fair confidence that you're not altering them. Great. I just have to go find this stuff, right? And it's all over the place. It's a little more pricey, but you can go find products that are, say, USP grade purified cotton. Okay, good. I can go get the right cotton. I can clean my teeth. I can feel like I'm not ruining them. Let's go look at some papers, right? Let's go look at some of these methods papers that I'm supposed to be following to find out what my rhinos eat. And let's see what vibes are not checking out. So let's look at the first one. The occlusal surface of each tooth was cleaned using acetone, followed by alcohol. What did they clean it with? A toothbrush? Cotton? Shellac remover? I have no clue what they did. That's not good. That's not good methods. So the method section of that paper, the steps that I'm supposed to be following, it's like a recipe. If you have a bad recipe, you might not know how the quantities you're adding. You don't know the time they're supposed to have that pie in the oven. You don't know what temperature. There's a lot of unknowns that you have to figure out. That's not good if you're trying to tell someone else how to do your recipe. You need to give them very specific directions. That's not very specific. I don't know exactly what they did. So I go to the next paper. After thoroughly cleaning the surface of each tooth using cotton swabs soaked in ethanol. Okay, cotton swabs. What kind? USP, Walgreens. And you used ethanol. What kind of ethanol? There's different kinds of ethanol. There's 200 proof. That's very, very pure and doesn't have much water. Then there's not pure ethanol with some water in it. I don't know what they did either. I should go to the next paper. These recipes aren't good. I don't know my quantities. I'm going to screw up my thesis and not get a job or a master's degree. So go to the next one. Let's read it. There's a big glimmer of hope, right? Okay, they use shellac remover. They are on the paracone. They tell me what part of the tooth they're on. They told me the product. They even told me the brand name. It's Zip Strip. They use cotton swabs. Yay. And they even tell me what kind. Q-tips on wooden stems. Hospital non-sterile. Non-sterile is not USP grade. They just told me they risked the chance of using contaminated Q-tips with silica on their teeth. I have no guarantee of knowing what damage was just caused. If there was damage, if there was alteration, I don't know what just happened to that tooth. Oh no, I'm getting terrified. So I had to pivot. I had now just gone down a rabbit hole on how to apply different materials to cleaning teeth to get to the surface to figure out what these animals ate. And I was now more interested in the things I was doing to teeth and what it was doing to teeth than I was in the diet of this rhino. And so this is how I got into fossil preparation, was in trying to become a researcher, because I started to see some red flags that made me concerned about the research that I was doing. And even more so, what was I doing to that fossil? They didn't know 100 years ago what they were doing to that fossil when they put shellac on it. I don't know what I'm doing to it now and what science I'm ruining in 100 years. I don't know what technology they're going to come up with. And that terrifies me. That terrifies me to my core. That fossil was around for 15 million years. It won the lottery to come to me. It has all this data. It's a fountain of knowledge. And I just erased some of it. It lasted that long, and I carelessly said, okay, yeah, this is what I'm supposed to do, and I ruined that data forever. We'll never know. That's horrifying. I don't want to do that. I don't want to be responsible for that. So I want to know exactly what we're doing to the teeth and doing to our fossils and how that can impact science. So I completely pivoted to look at some other parts of teeth. I wasn't looking at the preparation. I was looking at the physical structure of teeth and different parts of teeth to see, okay, do these patterns of scratches and pits even match across the tooth? Because I didn't want to think about what had happened to these teeth the first time they had prepared it and what I had just done. I felt like I couldn't trust that, right? So I thought I'll look at things that are away from the scrubbing that I just did. I'll look at different textures. I'll look at enamel microstructure, the way that teeth are built. I'll look at chewing facets. I'll try and look at other things. And I did find a lot of variation. Every part of teeth, they had different pits and scratches, different chewing facets. So where teeth collide and drag, different pits and scratches. Everything looked different to me. That was a concern, but that was part of science. I was starting to test methodologies. I was saying, wait, we don't know how all of these variables interact with the conclusion you're coming up with this paper. I'm concerned about that. I don't know what we're actually concluding in some of these papers. I want to know more about the methodology. I want to test methodology. Right, so I want to test that methodology. I want to go look at those skeletons. I want to know what happened to those skeletons, the entire process before they became that fossil or that skeleton in a collection or a museum that a researcher is accessing. What are the different phases that a fossil goes through when it's going into the museum and research? One is the field work. You have to find the fossil and you have to get the hunk of rock out. Two, that's the fossil preparation. That's removing the fossil from the rock. That's my job. That's where I landed. That's where I love. And then the last phase is when we have that research specimen and we can come in and display it. We can access it. It's out of the rock. It's something that we can handle. These first two steps are where you can really ruin a fossil and lose a lot of data. So fieldwork, we have to find the fossil, one, and the fossil's been going through a lot when we go and find it. So we're looking at these big rocky areas without much plant. We know that that age of rock has fossils. It's the right age for different kinds of dinosaurs or animals. We go walk around and we have to be lucky enough to be in the right spot at the right time to see a bone coming out of the ground. So on the top right, that's a fossil hadrosaur bone coming out of the rock in Montana. It's really fun to find. You were the first human being who's ever seen those fossils. You get to unearth it and discover it. It's a really satisfying event. But you have to remove a chunk of rock from the ground with a delicate fossil. Everything that you're doing to remove that chunk of rock could do something with that fossil. That fossil just sat out and had sagebrush roots eating the fossil and getting the mineral, and it had cows stepping on it, and it had careless field workers sitting on it or dropping tools on it. That fossil has already gone through a lot, just sitting on the surface and just being collected. What is that doing for the entire process? We're working in our fields. So these are some examples of our quarries that we have out in Montana when we're out collecting our dinosaurs that we bring back to the Burke, like our tough slug Tyrannosaurus rex. We're digging, we have shovels, we have picks, there's chaos, there's rubble. What are we doing? What are we damaging? When things are damaged, we also go jump glue on it. We have to hold it together. We have a little more understanding of the glue that we're using now. We're questioning, wait, what's this glue doing when I dump it on the fossil? What's the use of this fossil? Should I be dumping glue on it? What's that going to happen? What's going to happen in the lab when I have to go remove that glue? We're starting to think more about what we're doing when we collect this data. And there's been a lot of change in the last 20 years in the way that we collect fossils because there's recognition that we need to make sure that fossil is collected correctly because the research end relies on good collection and good preparation. Fieldwork's fun. You get to take big crews. On the left, I'm sitting there working on a triceratops, and I'll talk about that quickly because as a materials and methods expert, which is what fossil preparation is, you can take those skills back into the field and you can help train field people. When I started my career and I'm going to museums, I'm being handed random things and I'm not told why I'm supposed to use it. I don't want that to happen anymore. And I recognize that it happens. It's happened at every museum I've worked at. So now when I go to the field, I'm applying what I know in the lab and the way that I know materials and methods work to what I'm doing in the field to make sure that fossil gets to me in the best condition. So in this particular picture right now, I'm reading the rock. I'm not just a materials and methods expert. I have a geoscience degree. My fossils come to me in rock. I have to have a very keen understanding of how that rock forms, how it moves, how it breaks. The fossil's in it. It depends on that rock holding it together. That rock has held it together for millions of years. It's done a better job than any human's going to do. I need that chunk of rock to come out solid. That particular rock that I'm working in, you can see a lot of fractures and breaking, and it looks kind of like mud cracks because it's mudstone. It's very loose. It falls apart. It doesn't like a lot of weight or pulling or tugging, and that's going to ruin the fossil. So right now I'm looking at that rock, and instead of worrying about putting plaster and holding that whole rock together right now, I'm concerned about the ways that rock is going to destabilize. So I'm inspecting it and I'm shoring up the rocks so the rock does not destabilize. When I look at that block, I see a big heavy block of plaster and burlap on one side holding the fossil. That's a lot of weight that's overhanging. I'm worried that's going to fall. So in that picture, I'm adding plaster bandages and I'm holding them in place underneath the fossil because my first concern is there's bone or tissue that could fall out underneath that. My next step is to add a whole layer that goes around the edge to hold that whole top so that that edge doesn't slough off. So fossil preparation is great, not just for research and figuring out how we're screwing up your fossils, but it's good for the field end as well to take our knowledge back and make sure that the fossil is collected in the proper way in the first place. Then we get to take this fossil to the lab, and that's where I get excited. That's where I get to do the fossil preparation. So what is fossil preparation? I call it the art and science of removing fossils from stone. And what do I mean by that? Well, obviously the fossil's in a chunk of rock and I have to remove the rock. That can be very destructive. I can cause a lot of damage. Any way I'm removing rock could cause damage to a fossil. Why do I call it art? So this is a contentious issue in fossil prep. And even up to a month ago, I was uncomfortable with calling this art. And it's funny because when I started college, I actually entered college as an art major, right? Well, why am I weirded out by calling preparation art? Well, art has this connotation that it's created, that it's made, that it's something new. I don't want that connotation with the fossils I work with. I'm not creating fossils. I'm not faking fossils. I'm not adding things to fossils that are going to make researchers confused. I want to make that fossil come to the researcher in its a natural, unmodified condition. I don't want to change anything. And art makes me think I'm changing things, I'm adding things. So up until maybe even a couple months ago, I was uncomfortable with the idea that fossil preparation is considered art, even though it's tactile and you're using your hands and you're using your eyes and you're making things beautiful. I didn't like the idea that I was changing things. And then a colleague of mine down in LA, Alan Zidiniak, pointed out that what is similar between fossil preparation and art is the understanding of the different materials that you're using and their properties. So an artist knows how to water down paint and get paint to flow across a paper in the way that they want, and how to manipulate textures of oil, and how to get canvases to absorb pigments at different places. They have an understanding of what those different techniques applied to what they're working on can do. And they know different brushes and how to apply it and different scrapers. They have an understanding of the different ways they can attempt to do their art. Fossil preparation is the same way, in that we need to have an understanding of the different ways we can apply materials to fossils, the different tools, the brushes, the dental tools, the sand blasters. And we need to have an understanding of what we're applying and how that can affect the fossil. What are different ways to water down glue? How does the glue affect the fossil? We're starting to understand what we're applying to the fossil. And that is a whole world of conservation in museums that isn't new. People have been taking care of artifacts in museums for 100 years. Most research on what we put onto fossils comes from anthropology and archaeology because people are most important. So there's been a lot of science into what are we doing to fix paintings, 100-year-old paintings, 500-year-old paintings. How do we take care of them? There's a lot of research on how you clean those paintings and how you take care of them and make sure they last forever so we can enjoy them in museums. A lot of that's published. The difference between what I'm doing and an art conservator is doing is the art conservator already has a painting to take care of. I have to get my painting out of the rock first to take care of it. There's a little bit of difference there. But I have a lot of already understood knowledge about these materials and methods from anthropology and archaeology that I can take back to fossil preparation. I'm going to show you a little video. So fossil prep. I don't know if you can hear that or not. Actually mute that. So I like to make little TikTok videos and time lapses to kind of show my work because it's also fascinating. It's fun to uncover things. I'm uncovering this for the first time. I'm the first eyes that get to see it. And I like to talk about the tools that I use to prep fossils. So on this left side, I'm preparing out a T-Rex tooth from a Triceratops quarry in Montana. And on the right is the T-Rex tooth that I have prepared out. I'm making very intentional decisions when I'm removing this tooth from the rock. I understand what this tooth is going to be used for. And I'm thinking long-term about if anyone's going to have to come fix what I did in 100 years ago, or in 100 years. Because again, I'm fixing stuff that was shellacked 100 years ago. I don't want to perpetuate a lot of tedious work. So the fossil preparation is very engaging. It's very fun. And then out of that fossil preparation, you get your museum displays, you get your fossil that you can handle and manipulate, and you get all the fun research. How did that animal live? What did it eat? I get to go see it on the museum now. Great. So here's a couple examples of papers that I got to be involved with in the last 6 years working at the Burke Museum. I've worked on specimens that got to go in all of these papers. It's exciting to know that what I'm doing is going to contribute to research and new understanding of animals. And we have our big animals display, right? We can go do all of our research. We need to communicate, right, with the researchers and with the field workers, and communication needs to go in every direction. Because as I found out in my master's degree, not all methods are good, and there's not a lot of understanding, and not enough people are curious about what they're being told to use and how that could be impacting what they're doing. So let's go into the fossil prep a little bit more. I'm going to put my science hat on and move away from my artsy creative mode. Fossil preparation. It's not just art and science. It's the procedures that we use in the field and in the lab to make an object useful. That top photo of that fossil, you can see there's a fossil in rock. I don't know what it looks like. I don't know the teeth. I can't take samples. I need to make it useful for science. That's fossil preparation. On the lower photo, I can see the teeth. I can count the teeth. I can see what parts were missing. It's more useful for research. So if you want to research something, you have to prepare it. What are some other components of my job, and what are some of the considerations I'm taking when I'm taking these fossils out of the rock? That was a slip of my tongue. I need to identify what it currently looks like. Is it damaged? How is it damaged? What's deteriorating? And I also need to think about the future, because I'm cleaning fossils that were decaying from 100 years ago, and I don't want to do that to someone in 100 years. So I have to think about what I'm putting on it, and is what I'm putting on it going to be good in 100 years? Is it going to ruin the research and the technology we come up with in 100 years? I don't know. So I need to select appropriate materials. And the way I do that when I'm considering approaching fossils and preparing fossils is I take a very conservative approach. I modify it the minimum amount that I have to. If I have to add glue, I will. But I try not to, because I don't know what those chemicals are going to do or impact research. So I try not to alter it by adding chemicals. When I'm physically taking the rock off, I try to pick tools that are super gentle, that hopefully won't ever touch my fossil or gouge my fossil, that shouldn't abrade my fossil. I don't want to damage that fossil. I want to get it out of the rock with no modification, which would be ideal. But everything I'm doing, I need to understand, is modifying that fossil on some scale, whether chemically or under a microscope. I need to keep that in mind when I'm making these decisions, because that glue that I put on there could ruin the research of someone trying to figure out what that animal ate. It's not good. I also need to, pick those materials that are going to last forever. Again, because those shellacked teeth were shellacked by someone. Someone had to take the time and pay the money to get that shellac. It's not a good use of resources. We don't want to be doing redundant things in museums. We want to keep progressing and be good with the way we use money and materials. So I need stuff that lasts forever. I need the papers that tell me that it lasts forever. I need to do the research into, what is this? What am I applying to my fossil? I also need to keep records. I need to write down everything I did to that fossil. Did I scratch it with a toothbrush? Did I put shellac remover on it? Did I use a USP cotton swab, or did I use a dirty cotton swab from Walgreens, or one that I found rolling around in my drawer? I need to document everything I did to that fossil. And then I need to keep that record in the museum with that fossil forever. Historically, there's not been a lot of record keeping with fossils. Most museums that you go to do research on, you say, hey, this fossil from 100 years ago, do you have the record of what they did to it? A lot of people weren't keeping records. So keeping records has become important. We keep records of everything that's happened to our fossil, and it goes into our database so that researchers can know what we did to those fossils. I don't know what happened to those rhino jaws I was looking at. I only know that they had shellac on them. I don't know how they applied the shellac. Was it a brush? Was it a toothbrush? I don't know how they cleaned the tooth in the first place. I could be looking at teeth that have already been scrubbed with all sorts of things, and the whole surface has been worn and reworked. I don't know what happened to that fossil. That's not good. So we need to be good about documenting and retaining records. And it's important to enforce that and encourage that at different museums, because different museums have developed and learned about methods and materials in different ways. And they're not all at the same place, right? I've been worked at museums that are using Elmer's glue. And there are others that have moved to archival glues, or glues that will last forever, or hopefully forever, or a lot longer than 100 years. Not all museums are at the same place. We need to communicate. We need to get the word out to the community. You need to start being curious and questioning what you're doing to that fossil and how it can harm science in the future. On our right is a mammoth tusk at the Burke Museum. That was 9 months of my life. And that was a lot of decisions and a lot of testing to come up with that. I had to ask a bunch of people who've worked with ivory, how do I do this without ruining this fossil forever or making it fall apart? Ivory is really delicate. That's not fossil. So we need to communicate with our community. We need to know what's happening. We need to know what we're doing to that fossil so that people can come to that museum and see that tusk that was found in Seattle in 100 years. It's art and science. I have to master those materials. I have to think about 3D. I have to collaborate with other people. I have to look out for papers. I have to read and research. It's a very dynamic job. There's a lot of things you have to learn. You have to know chemistry. I was terrible at chemistry. You have to do a little math. I'm terrible at math, but I'm good at the art part. So I do a lot of the art and the creative part. So in this photo, I've got a part of a hadrosaur pelvis being supported by sandbags because fossils don't like when the rock is removed from around them that was holding them together. I have the sandbags holding it together now. I'm creating a permanent structure to hold it together even longer, and that's that white thing on the right. That is a fiberglass cradle used with polyethylene foam. All the products I'm using should last for a whole long time, hundreds of years. That should never fall apart underneath my fossil. It should be okay. So I'm creating long-term solutions to hold that fossil and make sure it's good for science in 100 years. I have to know how to work with plaster. I can take those skills with plaster back to the field and help people in the field understand plaster because I've been there. I've been in the field. I understand how it works. Someone says you mix it this way and you go. Well, I don't know if things go wrong, how to fix it. I've not been taught that. I'm just told what to do. So now I can take my understanding of plaster in the lab and how to teach people in the field how to use that so that I get the fossil in really good condition. We do all sorts of things. We don't recreate, but we do modify sometimes. So on the left is a salmon skull that we were missing a big chunk of the skull. We made a plastic replica, we stuck it on there, and then I painted the fossil so it looked, or not the fossil, I painted the cast to look like the fossil. So there's art and science right there. There's a useful science or fossil for science that can study the parts of that skeleton that are preserved, but I've also made it look pretty for display. And then we have documentation of what part is fake and what part is real. I'm part of science and I'm communicating with my researchers consistently. Every time I'm given a fossil to prep, I say, what are you doing with it? What is its purpose? What is the goal? I don't want to ruin someone else's research or that data. So it's very important for fossil preparators to communicate with researchers and for researchers to communicate with preparators. What did you do to my fossil before I do my research? There's not a lot of communication back and forth. There's not a lot of understanding. You have to have that curiosity about what happened to the fossil to start going, oh no, bad things could have happened here. So on the left was a cool project that I got to do a couple of years ago, cleaning the chest bone of a 250 million year old proto-mammal. And on the right is my supervisor kind of tweeting out some fun updates about that paper. I let him know every time I see something weird. And as I'm cleaning off that fossil from the stone, those little yellow tags are pointing to areas that I noticed were different. Those are tooth marks from another animal. So I'm the first one to see some of these things and alert the researcher, hey, your fossil is doing something funny. You should maybe look into that. So research, right? Fossil preparation and a lot of sciences within the conservation world and the natural history preservation world are starting to really amp up what we do with these materials and methods and how they impact science. A lot of fossil preparators and conservators are publishing now, especially in the last 20 years, looking at what does that Q-tip do? What does that acetone do? What does that plaster you dumped on the fossil do? We're starting to tease out how these different variables can affect the science. But there's still a lack of communication and there's a lag of getting that science communicated to museums because we have papers that were made in 2012 and 2010 looking at solvents and looking how to apply gels to clean teeth so you can look at the microwear. These papers have existed for a decade and they're not being incorporated into modern papers that are being published this year. There's some lack of communication back and forth, and that's not good. So it's really important to me to start communicating to everybody who will listen to me, the public, my students, researchers, field workers. Hey, I want you to be curious about what you're doing because that could affect someone. I don't want you to do what you were told without asking why. Why were you doing this? This is part of science. Asking why is part of science. Documenting is science. You need to be curious in science. And why is science limited to what we can figure out? Science shouldn't be limited to that. We should be knowing what we can figure out and how we figure that out. So these are some examples from those really cool papers on looking at the way we are affecting and modifying fossils. And it's useful because now I know, is it safe what I'm doing or is it not safe or do we need to find that out? And that is how we get our awesome fossils at the museum. That is the component of my job, a part of my job, right? And it's really rewarding. It's really fun to be part of science. It's really fun to get to test different materials. And it's very interesting, right? It's a mystery that I'm never going to solve. Every question that I answer and gives me 10 more questions. And there's a million different ways that you can prepare a fossil. There's a million different brushes and tools and techniques and materials and solvents and glues. There's an endless supply of things for me to go figure out how are you affecting my fossil? And how can I help research and science better and make these fossils last forever so that in 100 years you can walk up to the mammoth and go, that's a really cool mammoth. I don't want that mammoth to fall apart in 100 years. So that is fossil preparation and a little bit of its impact on science and how paleontology is all connected. The fieldwork to the preparation to the science, and communication needs to go in every direction. Understanding of materials and methods needs to go in every direction. And you need to be curious and question when someone says, here, put this glue on your finger painting. You go, why? Why this glue? Why not another glue? And then you write down why you're picking that glue and not another glue, because that's part of the scientific process. And then you put that paper in your drawer forever, right? So someone else can ask, how did you make your finger painting? Or how did you bake this bread? What was the recipe you used? Documentation is important. Observation is important. And communication is super important. So I hope I left you with a little more appreciation for those fossils that you see in a museum. I hope when you go to that museum and you look at that mammoth or the stegosaurus, you spend a couple of minutes instead of a couple of seconds looking at that. I hope you recognize a whole lot of work had to go into making that fossil for you to enjoy. I hope you enjoy it just a little bit longer, and I hope you go see your local museum soon and start asking questions about what did they put on that fossil? How did they get it up there? So thank you.